Personalized Medicine Center of Excellence is aimed at opening and explore frontiers to treat diseases at the individual level, combining clinical and omics information. To this end, PerMedCoE is focused at designing and developing workflows composed by building blocks. The challenge of PerMedCoE is to develop a sustainable roadmap able to scale up the existing software for cell-level simulation targeting European HPC exascale systems. Consequently, the objective is to provide a methodology to translate omics analysis into actionable models of cellular functions of medical relevance. With this goal in mind, PerMedCo is going to design and develop a set of building blocks ready to be used to compose complex workflows targeting cutting-edge medical research use cases, such as cancer diagnosis based on omics information, drug synergies for cancer treatment, tumor evolution based on single-cell omics and imaging, COVID-19 multiscale modeling of the virus and patient's tissue, and personalized modeling of groups of rare disease-related patients. The lessons learned and results from this process will be publicly available for the community. In particular, the idea that PerMedCoE pursues with regard to workflows focuses importantly in the existence of an efficient orchestrator able to process a user query in a computing infrastructure of pre-exascale or exascale size. This orchestrator deals with the available resources and has to deal with the execution of specific parts of the workflow. These specific parts, like personalized models, simulate, or validate, represent units of work that must be methodically managed across the computing infrastructure. In addition, the orchestrator has to be able to deal with complex infrastructures, and one of the key functionalities is to be able to exploit the joint power of multiple HPCs from different locations. Consequently, the user who wants to simulate drugs in patient-specific models can be using large amounts of resources transparently and seamlessly, avoiding to deal with complex management tasks. Being more precise, PerMedCoE envisions a two-level orchestrator, one located on top, responsible of dealing with different HPCs, and a second workflow orchestrator located on each HPC, responsible of the parallelization inside each supercomputer. The key in the workflows within PerMedCoE is the building block. A building block is a representative unit of work, like, for example, personalize a model or doing a simulation with specific parameters. It is written purely in Python for simplicity, portability, and compatibility. They are the bricks that can be used to compose workflows, and the PerMedCoE building block definition has a clean and precise interface which is composed by inputs that can be files, directories, or even objects, and a configuration file. The configuration file is intended to wrap all the configuration parameters for the building block in YAML format. A building block can produce files, directories, or objects as output, and this output can be the input for the next building block in a workflow. A building block has two main usages: as command line that can be invoked directly or imported and invoked from a Python code. Building blocks benefit from the advantages of containerization, such as portability, consistency, and security, among others. The execution of the binary or code included in a building block will be executed within a container, ensuring that all dependencies are met. The internal structure of a building block is simple, although can be complicated by the developer as needed, but the basics are really simple. For instance, all building blocks rely on the PerMedCoE package, which provides multiple automatic features like, for example, the execution within containers, the binary definition, and the task definition. The command line entry point is defined by the invoke function. This function needs to be tuned by the building block developer in order to convert the configuration file into the actual building block operation parameter. The main core of the building block is defined in another function, decorated with a set of decorators such as add task, add binary, and add container. This decorator stack enables to define the peculiarities of the function parameters, if it is a binary, and the container to use. The function parameters represent the signature of the binary to be invoked. The task can be pure Python if using a workflow manager that relies in Python, such as PyComs. This is not supported with workflow managers that rely on specific language definitions, such as SnakeMake and Nextflow. Finally, since the building block is written in Python, it can get as complicated as needed, and the developer can include as many tasks as required with an algorithm in the invoke function. This enables to exploit inner parallelism within the workflow when using PyComs. A building block is contained within a Python package that can be easily uploaded to PyPy and installed with pip. The PerMedCoE package provides the command line interface for the building block, making it homogeneous for all building blocks, and enabling the usage of building blocks within all workflow managers. The command line interface also has some advanced flags for tuning some of the execution peculiarities, such as debug level, container mount points, or requirements. However, the usage from Python as library is also valid for workflow managers like PyComs. As previously introduced, the PerMedCoE package has been developed to leverage users from the building block execution and management thanks to the decorators. It provides an homogeneous command line function and also has a unified interface for the workflow managers. In addition, the PerMedCoE package provides a tool to ease the application execution the single building block execution to create a building block template, which creates a complete Python folder with all initial results to develop your building block, and it also indicates in which files and lines you need to include your building block logic. And to create a workflow template for PyComs, SnakeMake, or Nextflow, which is particularly useful to start developing your workflow. So we have the bricks. These bricks can be used as many times as wanted and tuned with the configuration files. The next step is to connect them in order to build workflows of building blocks. In the scope of PerMedCoE, we envision a global orchestrator named Croupier to distribute the workload among high-performance compute, and three main workflow managers able to execute the workflows of building blocks within each HPC. A user can request the execution of an application written using Croupier, and the units of work of these applications are workflows, which may be executed in one or more HPCs, where the workflow managers will parallelize the building blocks among the compute nodes, respecting the data dependencies among them. SnakeMake and Nextflow workflows are defined via specification languages, based in rules or processes respectively. In the rules or processes, the building block developer must identify the inputs and outputs as well as the binary to be executed. Consequently, a workflow of binaries can be isolated. On the other hand, COMS, and in particular using its Python binding, known as PyComs, the workflows are defined directly in Python code, and the tasks and their peculiarities are defined with the @task decorator. In addition, PyComs can make use of the building block directly importing it and calling its method, exploiting the inner building block parallelism. Considering the three workflow managers, PyComs provides a set of characteristics that make it excel among them. For example, PyComs enables to speed up the workflow development since it does not require to learn a DSL, just Python. In addition, it is based in sequential programming, meaning that you write your application without worrying about parallelization and let PyComs deal with it. It is based in pure sequential Python, with a minimal API for the task identification, data directionality, and task types. The task is the unit of work, and consequently, since PyComps relies on decorators, a task can be any function decorated with the task decorator. In addition, PyComps provides a wide set of decorators to define binary tasks, const tasks, MPI tasks, and many more interesting features, such as executing in containers, polymorphism, constraints, etc. PyComps provides a simple linear address space of all resources of the infrastructure, enabling to run large memory-consuming applications. In order to perform the parallelization, the comps runtime builds a task dependency graph at runtime, based in the data dependencies among tasks, and deals with the scheduling, distribution, and data management across the infrastructure transparently to the user. Consequently, provides agnosticity to the users by enabling them to run the same exact code independently of the underlying infrastructure. In addition, PyComps has an interesting ecosystem composed by a monitor where the user can see the execution behavior, integration with Jupyter notebooks, and the performance trace generation for performance analysis. Building blocks and workflows play a very important role in the PerMedCoE project, where, for example, we have identified a set of tools that are wrapped into building blocks, such as Mabos, PCCell, Selenopt, and Cobrexa, and are key to compose the workflows that are necessary to tackle the project use cases and produce an impact in the community. Our pilot workflow is targeting COVID-19 disease. In this use case, the workflow starts with a Boolean model, which is evaluated many times using Mabos in order to determine which genes are activated, filtering the unnecessary ones. These are the initial candidates. In parallel, the single-cell processing prepares the input dataset. All these outputs from Mabos and single-cell processing are inputs for all evaluated patients, and for each patient we envision a preprocessing stage, followed by a set of PCBOS executions for each gene and a number of repetitions, finishing with a post-processing stage. PCBOS, which is the combination of PCCell and Mabos, performs the simulation of the cellular tissue to evaluate the effect of a Boolean model on each time step, which conditions the next time step and consequently the cell and tissue evolution. The workflow is finished with a meta-analysis stage. In this stage, we envision algorithms like PCA or clustering to extract the conclusions from the whole workflow. As you see, the potential parallelism is very high. We are not alone in this challenge, and for example, building blocks and workflows also play a major role in the BioExcel Center of Excellence. For example, they are also targeting COVID-19 disease, focusing on the infectious mechanisms and host selection process. In this workflow, they make use of a set of BioExcel building blocks to create a workflow that has been parallelized with PyComps successfully. The performance analysis of this particular workflow shows a good speed up thanks to the parallel execution of the BOBBs with PyComps, demonstrating that the building blocks and workflows are the way to go to scale up these scientific challenges and exploit efficiently the computational resources. Thanks for watching.